All right, now we're going to talk a little bit more about transporters. But first, I really want to have you kind of take a look at this graph and tell me which of the lines you think would correspond to movement of ions through a channel. All right, now that you've answered the first time, let's talk a little bit more about what we're looking at here. So here we have the x-axis, which is our independent variable. So that's the solute concentration difference. So in other words, the difference between the amount of solute that you would have on each side of the membrane. Right? And then on the y-axis, we have the rate of solute movement across the membrane. So how fast is that solute going to move across the membrane? And then we have basically these four lines, A, B, C, and D, that give a relationship of as you basically increase the gradient, right? More high, more low, as you increase that concentration gradient, how that affects the rate of movement across the membrane. All right, so now with all that in mind, let's go ahead and answer again. If you still have the same answer, that's perfectly fine, but see what you think the lines would be this time. All right, so let's take a quick look at this here. When you have a bigger difference, all right, higher to lower, things are going to naturally move faster, right? And if you open a channel, we already know the channel's fast, but think about it. If I have a lake, a dam blocking the lake, and that lake is very high compared to the river underneath, that's a high concentration difference. That would be somewhere over here on our graph, a higher solute concentration difference. And if I open up that dam, water is going to flow very, very quickly. And it's generally going to flow a lot more quickly than if the lake level was barely higher than the river level. Then I open the dam and it's going to flow a little bit more slowly. So essentially, as you get a bigger gradient, a bigger difference from the high concentration to the low concentration, you're going to have higher rate of movement across the membrane. Right? So that eliminates C right away, because basically in C here, as you have a higher concentration difference, you get slower. That wouldn't make sense. Now, we also know that channels move very, very rapidly. So when you open a channel, the ions and the solute can move very, very fast across it. So that probably eliminates D, right? And we know that basically, if you don't have any concentration difference, you're going to have no net movement, right? So that's what eliminates B here. That's the big thing here. When you eliminate B, or when you look at B here, even when there's no concentration difference, we still have movement across the membrane. And that wouldn't happen because there is no high, there is no low. So the one that makes the most sense here is A. And you can see that when you have a concentration difference, if you open the channel, it's going to move pretty rapidly across it. Now, what I want to ask you here is, why do we get this plateau? Why do you think we get a plateau at the top here, where even though we're increasing the concentration difference, we're not really increasing the rate of flow across the membrane? Well, the reason is because this is facilitated diffusion. These ions need help moving across the membrane, which we know because this is a channel. The channel has to be there to open up to create that pore through which ions and solutes and so forth can flow through. So if it's a sodium channel, the sodium channel has to be able to open up to allow the sodium to flow through, because the sodium can't get through the membrane by itself. And what that means is eventually we run out of channels. So at this point, even though it's very, very fast, all the channels are open, ions are flowing as fast as they can. It doesn't matter if you increase the amount of difference between the lake and the river. If all of your dam doors are open, then the water's flowing as fast as it can. So in this case, if all of the channels are already open, the ions are already flowing as fast as they can. So this plateau here is basically showing you that it must be facilitated, because you can maximize the rate because you run out of extra facilitators, you run out of extra membrane proteins, in this case channels, that would move things across. That actually is what really eliminates D here, because I don't really have anything that indicates rate here. You would assume that a channel should be really fast. But here, this never plateaus out. So if something is moving across the membrane on its own and doesn't need help, then the more difference you have in concentration, the faster it's going to be all the time, because you're not going to run out of helpers getting them across. So D would actually be your passive diffusion, things flowing across the membrane on their own, and A represents movement across through a channel, because you have rapid movement. The higher the concentration difference, the more faster flow you have. but you also get saturated where eventually you can't flow any faster because all of the channels are already open. Things are already flowing as fast as they can. All right, so now let's talk about transporters. So that was a channel. Well, what happens with a transporter is you have to bind to the transporter, and then the transporter has to change conformation and open to the other side. So essentially what's happening in the transporter, and take a quick look here at the video. So basically the transporter has to be open to the outside, and then your solute is going to bind, and then once it binds it has to open to the inside. So you need this conformation change basically to bind, change, and release to the inside. Bind on the outside, release on the inside. Or it could bind on the inside, release on the outside. Remember, with the transporter, it's still facilitated diffusion. All right, so based on that facilitated diffusion, which direction are my solutes, the ions or the molecules that it's transporting? Which direction are they going to go? Well, remember, facilitated diffusion is still diffusion. It's still going from high to low. So even though my transporter isn't like a channel where it just opens and things flow, it has to bind, change conformation, and release. It still is only binding to things on the higher concentration, releasing them on the lower concentration. It still is only moving things, helping things move, from high to low. Right? So which molecule is most likely to require a transporter? Water, potassium ions, sodium ions, glucose, or triglycerides? Well, the answer here is glucose, all right? And glucose would need a transporter because it's polar, so it needs help getting through, and it's bigger, all right? A triglyceride is nonpolar, so it doesn't need help getting through. It can get through by passive diffusion. And these ions, and even water, they're more likely to be able to go through via channels. But the reason glucose can't go through via a channel is because it's too big. If you had a channel pore big enough to flow glucose through, despite all of those things that we talked about with charge and size, exclusion, and so forth, a channel could not be specific to just glucose. If you opened a glucose channel, tons of ions, water, everything would flow through as well. So basically, things that need transporters are polar molecules that tend to be relatively large, too large to allow a channel to be specific. So now the reason you have specificity is my folded transporter here will have a binding pocket that's perfect for glucose or whatever it's binding, and then it'll release it on the other side. So a transporter needs to be this way because you never have just a hole big enough for the molecule to go through. You actually have to bind it on one side, then change conformation, release it on the other side. And so again, the things that most likely need transporters are anything polar or charged that needs help getting through, but generally the larger things for which a channel would not work. Because the larger things, you can't make a channel because too many things would flow through it. So then, let's now go back to this graph here, and you'll notice that I changed B. B no longer starts up here, so A and B both kind of start down here where you have no net movement across the membrane when you don't have a concentration gradient. But now let's assume that A corresponds to my movement through a channel. Which line do you think would correspond my movement through a transporter, the glucose transporter? Well, in this case, remember, a transporter is most likely to move things more slowly than a channel. A channel just opens up and the right thing will flow through. So if it's a sodium channel, sodium ions will flow through, but they're pretty much just flowing through. Just like you opened the doors at the dam, water is going to flow through pretty easily. You don't have to bind to the water, change conformation, release it. A transporter, on the other hand, has to bind to its solute, maybe glucose, and then change conformation and release. So bind, change, release, bind, change, release. And that's going to be quite a bit slower than a channel. So the answer here would be B, because you're still going to have a faster flow rate the higher the concentration difference is, right? And you're still going to see a plateau. So why would we still see a plateau where even though our concentration difference is getting higher and higher, where it still doesn't increase the rate at which things are moving across? Well, just like a channel, you can run out of transporters. So at some point, the transporters are all active and transporting as much as they can. It doesn't matter if you have more concentration difference, the transporters are already doing all that they can. So that's the reason for the plateau there. So Which line, A, B, C, or D, would correspond to passive diffusion? Well, the key to look for passive diffusion, remember passive diffusion, passive means it doesn't need help. So these would be things like the nonpolar molecules that can get across the membrane on their own. Diffusion, still from high to low. The key to know passive diffusion is that it never has a plateau. It never saturates because you don't need help, and therefore you don't run out of helpers. It also tends to be slower than transport, than things that get help either through a channel or a transporter. But the big key here is that if you don't see a plateau, it's because basically this straight line, the bigger the concentration difference, the more things are going to make it through the membrane on their own. And since they don't need help, they're not going to run out of helpers, as happens with channels or transporters. All right, and so again, just kind of to show the transporter, this would be a glucose transporter. You can see at one point it's open to the outside. Glucose will bind, and it's specific because this transporter has a binding site that only fits glucose. Glucose fits, fold equals function. The right interactions allow glucose to bind, then it'll open to the other side and release it, and then it'll basically do that again. So transporters, they're going to bind to the right solute because of size, but also because of hydrogen bonds, the right ionic bonds, kind of all those things that allow for quaternary structure or allow for a active site to bind to a solute. Those interactions are going to be very specific for one type of molecule. So again, a glucose transporter will only transport glucose. Okay? And it is slower because it requires that conformation change. That makes it slower than channels, but faster than passive diffusion. And it also means that eventually you'll plateau where you can't move anything more across, you can't move it any faster because you start to run out of helpers, you run out of the transporters. All right, so there are several types of transporters. They're basically completely based on how many molecules it is transporting and the direction. If it's multiple molecules being transported, the direction. So the number of molecules can either be a uniporter, all right, uniporter meaning it's transporting only one thing at a time. A glucose, transporting one glucose molecule at a time, would be a uniporter. And then the direction, if there's more than one thing, then are those two things being moved the same direction or in opposite directions of each other? So a symporter are two things moving in the same direction. So here's our example here. I've got these red molecules and the blue triangle molecules. They're both being moved from outside to in. Same direction, that's a symporter. Whereas an antiporter, they're moving in opposite directions. So in this case, the blue solute is moving inside, and then the red solute binds and moves to the outside. Opposite directions, antiporter. Okay. All right, so let's come back to the whole idea of potential energy. So we've been talking about water in the dam. When I open the dam, water is going to flow naturally from high to low, and this actually releases energy. In fact, you can have hydroelectric dams that allow electricity to be generated, energy to be generated by harnessing the energy caused as water flows from high to low. And that's the kinetic energy. The movement of water creates energy. Right? So again, things automatically go from high to low, that's diffusion. If things go from high to low, that releases energy. But if you're moving things from low to high, that means it takes energy. So if I was trying to pump water from the river into the lake, the idea of a pump right there means it needs energy. I need energy to pump water from low to high. And so just like water releases energy when it goes from high to low, it takes energy to move anything from low to high. And so which direction would sodium travel if a sodium channel opened? Well, sodium is going to travel from outside to inside because you can see I have a higher concentration of sodium ions in the extracellular fluid and a lower concentration in the cell. In the cytosol, so it's going to flow from out to in, high to low. What about chlorine? Well, chlorine also is going to flow from out to in. So the chlorine ions, when a channel opens, it's going to go from high concentration outside to a lower concentration inside. What about potassium? Well, potassium would flow from in to out. There's a higher concentration of potassium inside the cell, a lower concentration outside the cell. So if I open a potassium channel, it's going to flow from high inside the cell to low outside the cell. So those gradients are very important to cells. So remember how I said at one point, if you reach equilibrium, it's basically because you're dead. So the cells need to maintain a concentration gradient where there's higher sodium outside the cell or higher potassium inside the cell and so forth. So how do cells maintain these gradients? Well, that leads us to our last aspect of membrane transport, active transport. Right? So active transport is basically anything moving from low to high. And because it's moving something from low to high, so this might be pumping more sodium from inside the cell to the outside of the cell to make that concentration where you have the higher concentration outside. So anytime you're moving something from low to high, it requires energy. And energy usually is in the form of ATP. So which of these processes here, A, B, C, or D, which one is showing an example of something moving something against its gradient? So when I say against its gradient, I mean moving from low to high. Well, D would be something moving something from low to high because it needs energy. So your key aspect here is if it's moving something from a low concentration to a high concentration, it needs energy. Anything moving from high to low doesn't need energy, whether it's passive or facilitated. It doesn't need energy if it's moving from high to low. In fact, it actually gives off energy. Right? So active transport is the movement of something from low to high, and it requires energy. All right, so here we have our passive diffusion. Right? So this is a good review. Passive diffusion, it's going from high to low, and it doesn't need help because these are non-polar. Facilitated diffusion, going from high to low and does need help, either through a channel or a transporter. And then active transport is basically anything moving something from low to high requiring energy.